by lipids, the compounds that we would normally describe as either fats or oils. And sometimes we'll tend to use those words rather than lipid. But lipids covers fats and oils, and the only difference between those is the state that they're in at room temperature. A fat is solid and oil is liquid at room temperature. That's it. So we will sometimes say fats and oils, we'll sometimes just say fats. What we should say is lipids. Lipids covers a sort of broader range than just what we're familiar with as fats and oils as well. There are also a lot of compounds related to lipids, and what I haven't included in the notes for this one, I realise, is any of the functions and the related compounds. So I'll need to just add a little bit at the end on this, I think. The thing with lipids is that they're much more variable than the carbohydrates, so we can't just give them a general formula. With the carbohydrates, we had that CXH2OY showing you that for any given number of carbons, you'll have a number of hydrogens and oxygens that are in the proportion that you find in water, hence the hydrate part of the name. Essentially carbohydrate just means hydrated carbon, doesn't it? Water and carbon. That's not how the molecules exist, obviously, but with lipids we've got carbon, hydrogen, oxygen again, but they're not in any predictable proportions necessarily. It depends on the type of lipid that you're looking at. It will vary. So carbon, hydrogen and oxygen again, but what they do have a lot of is carbon and hydrogen. So you get hydrocarbon chains in lipids. Now, lipids are not polymers. They are macromolecules, large molecules, but they're not polymers. Right? So that's quite significant. A polymer molecule would be one that's formed from repeating chains. Sorry, repeating units making a chain. Whereas a lipid is not repeating units. Right? They can be very, very large molecules, but they're not made from repeating units. There are two main types of lipid that we're going to consider, which is just ordinary triglycerides, we call them, and then the phospholipids as well. Now, you're probably aware of molecules like, say, cholesterol. We've mentioned steroid hormones and other substances, haven't we? And those are related to lipids, but they're not actual lipids as such. They are lipid soluble, so we talked about the steroid hormones being able to pass through the phospholipid bilayer of the cell membrane, didn't we? But they're not something that we can draw structurally in the same sort of way. So the first bit then, the triglycerides bit, we've got two component parts to a triglyceride. And if you ever have a blood test that's looking at your lipid levels in your blood, I mean, they look at the cholesterols, there are two sorts of cholesterol. You've got the high density and the low density lipoproteins. They look at your total cholesterol, but they also look at total triglycerides. So they will look at the actual sort of fat content of the blood. So the two components we've got here, you can see drawn out without their bonds. You've got glycerol and you've got fatty acids. Now, the reason it's written as glycerol is because this is biology, not chemistry. All right. So first of all, the glycerol molecule. What's the actual chemical name for that? Does anyone remember? It was in the assignment, you're absolutely right. It was propan, yeah, for the 3. And then it's 1, 2, 3, triol, yeah, all right. So, yes, I shan't bother calling it that necessarily, the other group, but, you've got this overlap area that gives you a bit of an advantage. So glycerol then it's an alcohol. It's got 3 OH groups, and you should be able to put the bonds into that skeleton molecule there without any trouble, shouldn't you? I've drawn it this way round for convenience, but don't assume that it will always be drawn this way when you see it in questions or textbooks or anything like that. All right, with the OH groups coming off the right-hand side. I seem to remember when I did this, when we were doing the alcohols, I did it just before Christmas, did it the other way around, and we got ho, ho, ho down the side. All right, so obviously if you flip that over, it's over here. But the reason I draw it that way round, with its hydroxyl group sticking out to the right-hand side, is because when we join on the fatty acids that are going to be involved, it's easier to draw those on the right-hand side, and they are going to react with the OH group, so it's just easier to have them there, position-wise. So this is actually a very small molecule, glycerol. It's only got 3 carbons in it, and it forms a very sort of insignificant part of the molecule, really. It's not a part that we really pay very much attention to, but when fats are broken down in the body, or they're broken down for use, then the glycerol will be separated. We can use that for energy. It's about the only thing we can use it for. I suppose we can convert it into other things, but the actual sort of function of it in this sort of molecule is really to hold the rest together. The other parts are far larger. We're going to have, in a triglyceride, immediately tells you how many fatty acids will be. 3, yeah. So 3. We're going to join 1 onto each of those OH groups. And it's a reaction essentially between an organic acid, a fatty acid, we call them in biology, we'd call them carboxylic acids in chemistry. We're going to have 3 reactions between an alcohol, because there are 3 OH groups, and the 3 acids. So we've actually got 3 reactions that go on to create our triglyceride. And the type of reaction, it's esterification. It produces ester bonds at each point, because an acid and an alcohol will react together to produce an ester. I don't know if you remember, we had a look at some of the smells that were related to esters, particularly fruity smells, floral smells, but also that's not relevant here. There was all sorts of smells like goat and petrol and all sorts of, they were general sort of organic smells we were talking about at the time. But the glycerol molecule itself then provides us with these 3 sort of reaction sites, if you like, where we have 3 functional groups, the OHs, and so we can now join fatty acids on there by reacting 3 with those 3 OHs. The fatty acids, however, require a little bit more background. I will put carboxylic on here. All right, the fatty acids, or carboxylic acids, we know the functional group of a carboxylic acid is that CO, that's it, COOH bit. It doesn't matter which end we draw that on. When we start with this, I'm going to draw it, you can see it's coming up on the right-hand side of the molecules, just because that's sort of convention as to how we draw them, I suppose. But, of course, when we start joining these together, I'm going to have to flip those round so it's on the left-hand end, so we can react them with the OH group. So don't let that bit confuse you. The fatty acids are generally, I think I've put in here, 16 to 18 carbons long. So, in terms of proportions of the molecule, if you've got 3 of those, that makes the glycerol incredibly insignificant. Obviously, it's not insignificant because it's holding them all together, but what you've got is, mostly carboxylic acid. It's mostly hydrocarbon chain. When we use fats in the body for energy, then those hydrocarbon chains are a huge source of fuel, and they are chopped up into 2 carbon chunks that can just be fed into all the respiratory pathways, but we will come back to that. Right, so you've got one here which has 1, 2, 3, 4, 5, 6 carbons, because I couldn't be bothered to draw anything any longer. The time it took to line all these up, well, you can see there I've used the tab actually to do it. But we've got very simple, all you've got to do is just to remind yourself that if we're going to put all the detail in. Of course, we've actually moved away from this in chemistry and started doing the abbreviated skeleton formulas and things, haven't we? So 1, 2, 3, 4, 5. And then the sixth one, we have our carboxyl group on, the acid bit. If we were going to draw that as a skeleton formula, you'd have the zigzag, wouldn't you? Yeah, to represent the hydrocarbon chain. Now that we'll come back to, because that's often how it's drawn on the abbreviated form of a phospholipid, particularly when we're talking about cell membranes. A little way off for that, yeah. But we know that that thing doesn't form a straight line. So what carboxylic acid is that one? Very simply. How many carbons? 6, so it's hexanoic acid, yeah, hexanoic acid. Too small to be actually part of a triglyceride, but like I say, it was more about space, and you know, you didn't want to really be drawing in all the bonds between perhaps 16 to 18 different carbon atoms. Now, what we have here is a saturated fatty acid, and we've met the idea of saturated and unsaturated molecules in chemistry anyway, haven't we? So, you know, so I say you're at an advantage. The way I used to have the course designed was so both groups had done that much chemistry before we got to this point. The other group haven't actually. They haven't touched organic chemistry yet. So I think I need to redesign because they'll find this quite hard. So this idea of a saturated fatty acid is because we have only single bonds between the carbons. Yeah. So single bonds between the carbons gives us a saturated fatty acid, that one being an example. And saturated fatty acids will therefore give rise to saturated fats, which of course are supposed to be evil, aren't they? They've gone through a phase of being evil. They're now less evil than they were. And there is now quite a move towards introducing healthy saturated fats back into the diet, you know, that we were supposed to be trying to remove them altogether. But this is one of the things in science that you've got to keep your eyes on, and particularly anything that relates food to chemistry and vice versa. You know, the actual evidence and all that, it changes. You know, you'll find so many studies have shown this. Oh no, so many studies have shown that. Oh, so many. Oh, everything does. Yeah, everything. You know, eating this raises your risk of heart disease. Eating that reduces your risk of heart. Oh, it happens to be the same thing. You know, drinking this much coffee, or you know, if the thing is, if you tried to follow every bit of advice, you couldn't do it. You know, there's a certain degree of common sense. But if you're thinking of sort of making particular changes in your diet, look for sort of proper evidence and think about the biology of it, because there is some absolute utter rubbish out there. The saturated fats thing, I don't see that saturated fats should be seen as a baddie. You know, if you think about it, what would be the original source of fats in a human diet? Probably animal products, wouldn't it? Because plants on the whole don't contain that much lipid. Yes, perhaps from grains, but you have to, if you go back in our history, you know, there's not going to be that much use of grains, is there, early on? I suspect animals were eaten before grains were cultivated, you know, before agriculture. So, you know, our diet would have contained animal fats. And then we were talking about milk yesterday, weren't we? And the idea of dairy fats is actually, you know, we're coming back to thinking that those are healthier, and oh, you know, you've just got to, yeah. I would say take it with a pinch of salt, but I'm not sure salt's good for you. So that's our saturated fat anyway. It doesn't, you know, the fact that opinions are changing doesn't mean you should, you know, all of a sudden go out and start eating, you know, butter for lunch or something like that. Deep fried butter at that. With chips. The other option, of course, is you can have unsaturated fats. Now, they come in different varieties. You could have a single double bond in the chain, in which case it would be a mono unsaturated, or you could have two or more double bonds, in which case it would be polyunsaturated, yeah. So when we talk about polyunsaturated fats, that's because they've got polyunsaturated fatty acids in them, and that just means 2 or more double bonds in there. So what have we got for that one? We've got 1, 2, 3, 4, 5, 6. Ooh, 8. All right. Let's see if we can draw that one. 3, 4, 5, 6. Whoops, done them too close together. 1, 2, 3, 4, 5, 6, 7, 8. Now, where's the double bond going to come in that? We've got hydrogens all the way along here. And then we have these ones. 1, 2, 3, 4. And then I've got one there. So I'm going to need to put a double bond. Well, there, aren't I? Yeah? So that all our carbons have now got 4 bonds. So there is our unsaturated bit. Now, that's a little bit misleading, and we probably ought to get the model things out at this point and show what it does to the shape of the chain. But double bonds, I can't remember whether they put a bend in the chain or whether they straighten it out. But the shape of the fatty acid is different if you've got a double bond in it. And the arrangement of the hydrogens either side of that bond makes for different sorts of fatty acids as well. Have you heard of trans fats? Yeah, it's supposed to be really bad. I'm not entirely sure what which bit of it it's referring to, but you could, of course, arrange that differently. At that point, you could have, if you came in there, you could have your hydrogen there, and you could have 1 hydrogen up, 1 down, or 2 on the same side. Now you know from the modelling that that's actually different. That's in 3 dimensions. Because you can't rotate around a double bond, those are going to stay in those positions. Now, the idea of trans means sort of on opposite sides, if you like. So this is a trans arrangement here. All right, where they're opposite. This one would be cis. All right, it's not something we'll come across anywhere else particularly. But yeah, the trans fatty acids presumably have an arrangement like that. The other thing that you have to be aware of with unsaturated fats, what's the general source of unsaturated fats? It's, just in terms of a type of food product, it would be, yes, the vegetable oils, so plant origins, the unsaturated ones. But you will sometimes see, if you're looking at ingredients on packets, because I'm sure you do, you will sometimes see hydrogenated vegetable oils, and you think, oh, it's got vegetable oils in it, can't be too bad. Hydrogenated, yes, that's important, isn't it? So if this is in a vegetable oil and you hydrogenate it, where's the only place we could stick more hydrogen? Is actually on here and here, isn't it? What do we create? Saturated fat. Yeah. So hydrogenated vegetable oil is it's a bit of a con. Let's rewrite that in there. So yeah, beware of hydrogenated vegetable oils. They are not unsaturated. They are saturated, and artificially saturated. That's the problem. Saturated fats that might come from, let's say, meats and fish, no, not so much fish, but dairy and all that. Not so bad, but yeah. Well, the saturated fats, the argument against saturated fats is the way that the body processes them. And there has been research in the past that's shown that a high level of saturated fats in the diet can lead to increased levels of cholesterol, and cholesterol can deposit in blood vessels and lead to heart disease. And it's just the way the body processes them. Whereas polyunsaturated fats don't raise your cholesterol, and monounsaturated fats can actually reduce your cholesterol. And it's about the way that the body transports these substances as well. So it's quite complicated. You know, it's not actually the individual bonds in them. But and it's you know, sort of a bit of a sidetrack. But at the moment, because I'm trying to get my blood sugar controlled by diet rather than having to go on the medication, I'm trying a diet which is high fat, low carb. So I'm eating virtually no carbohydrates, but replacing with fat. And there's been a lot of research into this, that actually you can actually lose weight by eating high fat, low carb. I'm not recommending it to anybody. And I did lose a bit before Christmas. I actually lost about a stone in the month up to Christmas. Then everything went horribly wrong over Christmas, and I've only just got back onto it. But it had also brought my blood sugar down as well. So it was actually working, but I hadn't had my cholesterol levels tested. So a bit risky on that point, and that's something that I'm back on it now, and I'll get my cholesterol tested, and I will tell you what it's doing. But it means that, yeah, I don't touch anything sugary apart from fruit, because I can't give up fruit. But I don't eat anything carby like bread, potatoes, rice, pasta, which it's all right once you've stopped for a while, then, you know, it's not too bad, but at first it's quite hard. But instead, my snacks consist of things like pork scratchings and cheese, which is great. And macadamia nuts, I think. So it's a diet that's it's got its strong points actually, and I've had to hunt down, because I can't give up chocolate completely. I've had to hunt down the lowest carbohydrate chocolate, and it turns out I think it's Lindt is the brand, and it's the 90% cocoa one, which is an acquired taste. But I have now acquired that taste. But I try and limit myself to no more than 2 squares at a time. But that is actually quite satisfying. So we'll see. I'm an ongoing experiment on this. But it will depend on what my metabolism makes of it, because everybody's metabolism is slightly different. And this is another thing with diets and fats and carbohydrates. Generally, not everybody processes them the same way. You know, we have genetic differences and just basic metabolic differences. Some people's metabolism runs faster than others. Other people can, you know, eat huge amounts and just never put on any weight, and, you know, I hate them. And, you know, but it's the idea of the standards of healthy diet for anybody, it just shouldn't, it shouldn't exist really. But yeah, I seem to think there was it was on my news feed, I think yesterday, that somebody was actually saying that a higher fat diet is not as dangerous as the government have been making out. But the high level, it wasn't the high fat thing, it was actually saying high intake of carbohydrates is not good for a lot of people. And it is the biggest contributor to type 2 diabetes, more so than obesity. It's the carbohydrate load. And yet, of course, government advice is that your meals should be based around carbohydrates. You know, the first thing you put on your plate is your rice, your pasta, your potatoes, that sort of thing. And then you add the other things. But, you know, that's not necessarily suitable for everybody. And you know, it's watch that space. And, yeah, fats are in the mix as well. So, so these are our fatty acids then. We've got the saturated option, the unsaturated option. And we now have to stick them together. We've got to react our fatty acids with our glycerol. And so triglyceride, we've got 3 fatty acids, where the carboxyl group, which of course is this bit. All right, so there's your carboxyl group, as on any fatty acid, carboxylic acid. That's the functional group, thinking back to chemistry. And that's going to react with the hydroxyl groups on the glycerol. So let's go back up and find those. All right, so these are our functional groups on here. So we've got hydroxyl groups. And so if we flip our carboxylic acid around and put the carboxyl groups sort of against these hydroxyl groups, we can then have a a reaction between them to create a triglyceride, and you can see on the next page of the handouts that gets distinctly messy. But if you look at it, you know, it's there to just to make you sort of process it, really. You don't have to draw it out, at least. All you've got to do is put the bonds in. And you've got three sort of panels in here almost. The top two are the formation of a triglyceride. So we start off with our, I have to draw, oh dear, that went wrong very quickly, didn't it? So let's do that before I get in a mess with it. I'm not going to fill in all the bonds. I'm just going to make sure that you get the joining bits right. Right, so that is the COOH group of our first fatty acid up here, and it then goes on to its hydrocarbon chain along like that. Then you've got the next one. Right, so just make sure I haven't left myself enough room here, really, have I? The next one is getting very squashed, and again. So we only really need the reacting bits here. Oh dear, that didn't. I could have just copied them off the Word document. Now, the reaction then is between the hydroxyl group and the carboxyl group, and common sense would tell you that an acid is a proton donor, a hydrogen ion donor, so we'd be taking the hydrogen off the fatty acids and the OH group off the glycerol. It's the other way around for some peculiar reason. All right, and I have to sort of check myself every time I do this. But what we're actually going to do, it's a condensation reaction, ultimately, but in this case it's one that we've met before, and that's esterification, formation of esters. So we're going to remove the hydrogens actually coming off the glycerol. So we've got three places where that's going to happen. And we're going to produce enough bits there to make 3 molecules of water, yes. And so our condensation reaction down here, all right, so this lot, these 3 are coming out, so this is our minus 3 waters. We will put in, well, no, you've already got condensation in brackets, haven't you? But I would put, it is actually esterification, esterification. Not surprisingly, of course, you can put those back in like we had yesterday. Ultimately, when we've got our triglyceride down here, you can break them apart. And of course, when we digest fats, we hydrolyze them, we add water back in, break the fatty acids and glycerol away from each other. But what we're going to have at the bottom then is, all right, the glycerol we can just draw in to start with. But then, you know, if you look at what's been taken out, the oxygens are left from the OH groups on the glycerol molecule, so they have an unpaired electron now that needs to join to something. On the other side, we've lost an OH that was attached to a carbon, so again, we've got an unpaired electron there. So we can now just go to the O, and then we should get that. So that is an ester bond. Let's put a ring around that. Okay, this is our new bond here. All right, and that is an ester bond in this case. And so we will have 3 of those. And the fatty acids aren't necessarily all the same. They could be, but they don't have to be. So, so that's now got very distorted down there. But what we've ended up with is these 3 new bonds here. All right, so these are ester bonds. All right, so hopefully you can see where all the little lines go in that now. So the hydrocarbon chains don't have any role in this process. You know, they just sit there and they stick out at the side as great big non-polar chains, don't they? You know, consequently, they are not going to interact with water. Hence, triglycerides are not water soluble. You know, all that hydrogen and carbon there is, yeah, going to make them very insoluble, so they don't interact at all. And then, of course, we've got the phospholipids. I've not put the whole thing together to fill in all the sort of formation part of it, but the difference here is that one of the fatty acids, and you know, you'll always see it drawn the same way, it's the one at the bottom of the diagram, replaced by a phosphate group. Now, what I've drawn on there is not strictly speaking a phosphate group, but it works the same way. Basically, it's some phosphorus and some oxygen. It comes from phosphoric acid. So you have, so let's put the carbons in because that's where I get in a mess, isn't it? We've got, we do still have the hydrogen at the bottom. We've got our that for two of the fatty acids, but then we have this other arrangement down here where instead of the long hydrocarbon chain, we've got phosphorus. Phosphorus forms how many bonds? Any offers? No. Yeah, it is actually 5. I'm just trying to think. It's not necessarily obvious on there, is it? No, but it is. Yeah, to the oxygen. Yeah, you get a double there. It's got different valencies. It is a little bit of an odd one. You won't often see it drawn like that. You often see it drawn as an O minus and perhaps an odd hydrogen ion somewhere. It doesn't particularly matter. It's just the fact that, and if it was like that, the polar bits on here, of course, would be here and here particularly. All right, so those are polar. We know that electrons will be pulled more towards the oxygen than towards the hydrogen. So that immediately makes this, you know, a very polar molecule. That little bit is the important bit. So that when you draw a phospholipid, of course, you draw this polar phosphate head, and then the lipid tails are either drawn as straight lines or you'll see them drawn as zigzags. And so those now represent those, yeah? So now this makes a bit more sense, doesn't it? That those zigzags are these long hydrocarbon chains, and that polar head is this bit. Yeah? So all of a sudden, the first unit now makes more sense. So, yeah, that is your polar hydrophilic, if you like, but we can't put it all in. Polar phosphate head that will interact nicely with water, and then your hydrophobic bit, the non-polar. We often say, at that point, we say lipid tails because we haven't done hydrocarbons. But I'm going to put hydrocarbon tails here. So those are the two fatty acids on here, whereas the phosphate bit will interact nicely with water. You know, sticks itself on the outside of the membrane or the inside, well, you know, on the surfaces, shall we say, inner and outer surfaces. Okay, the one thing that I haven't included then in this, and I don't know whether I left it out on purpose when I was putting these together, but there are a lot of related compounds. The trouble is they do get a bit complicated because they don't look like this. If you look at things like the sterols, for example, although they are related to lipids, they don't have these long hydrocarbon chains and things. They have rings and stuff like that, which make them look different, but they originate from lipid molecules. But sterols, now you might have heard that word, and I've used it on purpose. Can you remember where you've heard it? Do you watch your adverts on telly? Sterols, plant sterols you find in, sorry? Possibly in, yeah, the Flora Proactive, possibly. It's Benecol is the one that advertises having plant sterols in, um, which help to reduce cholesterol, theoretically. But, yeah, I say theoretically, I'm never entirely convinced. I sometimes think that if you make that sort of change to your diet, you may well be making other changes to your diet as well. It's not necessarily the thing that you're taking. Um, Anyhow, yes, steroids, waxers, and things like that as well are also related to fats. You know, if you think what wax is like, beeswax and the waxy surface on leaves, related to fats, if only in their water repellent sort of nature. You know, you put wax on leather, don't you, to make it waterproof, for example. And so we have a lot of related compounds as well. Generally speaking, fats are used, well, lipids are used for a variety of things. Most obviously for energy, though, because they have this great long hydrocarbon chain that can be chopped up into chunks and fed into respiration. But also the waterproofing I've mentioned. I mean, we produce oils onto our skin and hair. If we strip all those off, then, the skin is very vulnerable. So, need to maintain oils, which is why we use moisturisers. Moisturisers, oddly, are just a way of putting oil back into the skin. We strip it all off and then we put it back on again. So waterproofing, we've also got things like the steroid hormones obviously rely on a lipid base. And yeah, so, uses like that. But the other use, of course, in animals is for insulation. And storage, energy storage as well. Unfortunately, some of us are very good at storing energy in the form of lipids, which is, it sort of goes back to the times when we didn't find food so readily, perhaps, that we would need to be able to store food for times when there was less about. Sadly, evolution has not adapted to the fact that we now have Tesco and Asda. Food is always available, isn't it? But food is very rarely not available to most of us. Well, I don't know most of us. I mean, obviously there are countries where this is not the case. So it's a bit blasé just to say, oh, all humans have got food readily available. But a large part of the human population has got relatively constant access to food. So we shouldn't really be storing it, because storing too much of it is not a survival advantage to us anymore. It's actually a disadvantage, because it can make us very ill. It can actually shorten our lives. So you would think we'd have sort of evolved perhaps to store less fat. We haven't. Maybe eventually, who knows. So uses for lipids generally, like I say, I've not included a sheet in the handouts for this, but the general sort of functions include a fuel, it's an odd way of putting it, but it is fuel for respiration. In other words, a good source of energy. Storage, and it's storage of energy, really, isn't it? Which is an odd way of looking at it, but that's what we're doing. If we're storing fats for later use, then when energy's required but we haven't got food available, then we should be able to turn to our stores. Insulation, particularly in animals, and then waterproofing, sort of across animals and plants. Not sure about other things. And then hormones. Off the top of my head, I can't think of anything else at the moment, so that's why I put include. The energy side of it is very significant in terms of the structure function thing is what I've got to keep coming back to, and it's the structure of having these great big long hydrocarbon tails that can be chopped up into bits for energy, because the little 2 carbon pieces can be fed into, remember Krebs cycle from the first assignment? Those 2 carbon pieces can go straight into Krebs cycle. They don't have to go through glycolysis first. So fats are a very ready source of energy. And the waterproofing bit, of course, relates to the polar, non-polar thing. The non-polar triglycerides are obviously going to repel water, so particularly good for waterproofing. Okay, I'm going to stop it at that point and name